And thank you to all the BAGIM organizers for allowing us to host today's event. So as this slide states, the topic of today's event is structure and ligand-based analysis and how to combine the best of two worlds for driving compound design. So this is what I had in mind for the session. So I'll start with a short introduction of who we are and why we've chosen this topic. And I've then asked our two panelists to share their point of view and some personal experience on the topic in a short presentation. And after that, I'll moderate a roundtable discussion between the three of us. And lastly, we'll open up the discussion to the entire audience in an organized way. So you need to print your questions, type your questions in the chat box, and we'll go through them one by one. So yeah, please feel free to type them as we go along, and we'll treat them at the end. So I hope this sounds good to everyone. So who am I and why am I speaking about structure and ligand-based methods today? Well, I'm Gabriella Jonasson, and I'm not based in the Boston area, but outside of Paris. And I work for a software company called Discngine, which provides scientific solutions and services to the life sciences industry. And over the last decade or so, we've worked closely with both medicinal chemists and CAD on different parts of the drug discovery, of the early drug discovery process. And what we see is that it's still not easy to combine information coming from the ligand-based methods with the information coming from the structure-based side, at least not in a manner that is efficient and consistent from project to project. Often where if there are structural data available in a project, aim is typically to exploit these structures to the fullest. But how do you in practice capture 3D information and combine it with information coming from SAR models, matched molecular pair analysis, and molecular descriptors in general? So from what we see, it's tricky and time consuming often. So I'm going to just take a concrete example of what I mean. So let's say you're in a project where you enumerate a library of compound ideas. So you have a couple of thousands of molecules. And of course, to save money and time, you want to reduce, to filter down the library to a few hundreds or even less before sending the molecules to be tested on your target. So to guide the selection, so that you keep only the compounds with the highest chance of becoming a lead, you want to prioritize the ideas or the compounds based on a combination of predicted properties. So coming from QSAR models or phys-chem properties, and also 3D predictors, such as the structure compatibility with the 3D structure of your target. So if this is what you want to do, the challenge often lies in gathering and formatting the data that you need so that the project team can take a decision quickly on what to push to in vitro testing. So I'll come back to this example later and other scenarios where these worlds needs to be combined to make the project more efficient. So I'll come back to that in the panel discussion afterwards. But to sum up things and finish my introduction, the main aim here today is for us to discuss together around this topic and try to identify best practices, some quick wins maybe, and also pitfalls to avoid. And yeah, let's see if hopefully we can get input from the audience as well. But first, let's start with our panelists. I have a new slide on this. Here we go. So the first panelist is Malin Lemurell, who is an executive director and head of medicinal chemistry in early cardiovascular, renal, and metabolism BioPharmaceuticals R&D at AstraZeneca in Sweden. Oops. And we also have Troy Smith as the second panelist, who is a senior expert in data science at Novartis Institutes of Biomedical Research here in Boston. So welcome to both of you, and I'll let Malin start. I'll reactivate your micro. Did that work? Yes, perfect.
Yes, thank you very much.
And I'll leave the screen. Yeah. I'll mute myself.
So I will just see it so I can share. This should be right. Share. So,
yes, I see. Yeah, I see the slide.
But it's not in screen mode yet,
Not
right?
yet, not yet.
No. Then we get it there.
Yeah, perfect.
Yes. Thank you, Gabriella, and thank you everyone for having me here today. As Gabriella said, I'm part of the AstraZeneca Gothenburg community team, and there I'm heading up the medicinal chemistry for the cardiovascular, renal, and metabolism, and also part of the global chemistry leadership within AstraZeneca. And I'm heading a department having medicinal chemists and computation chemists and synthetic chemists. And so I'm not a deep expert myself in computation chemistry, but I have done medicinal chemistry for many, many years and have strong experts in my department. So hopefully I can contribute. So first of all, on the topic, I just wanted to share the importance of the topic chosen here by Gabriella and Discngine around merging the understanding and insights we have from 2D structures and data generated with 3D. And then this is an example for one of all our projects on MR modulators, which we started out as following up on MR antagonists that were out marketed already when we started, based on the steroid scaffolds, where we did know that they had some very beneficial effects, but they were not possible to position into the cardiorenal patients due to the side effects of electrolyte handling and the risk of hyperkalemia. So when we started this project here, after some time, we generate some really interesting SAR to also be able to understand how we could find modulators within this series, which would block also the translocation on the nuclear hormone receptor, or actually wouldn't block the translocation of the nuclear hormone receptor into the nucleus, which we did see that the eplerenone or other spironolactone lactone steroids would do. So by that, we could demonstrate preclinically that we had a good separation of organ protective effects from the electrolyte by side effects here. And what we saw here, using a lot of crystal structure and insights, that we could really map out the binding site here with which part would trigger antagonism, which would trigger agonism. And there was also a super agonist pocket, which we saw, which really pushed the agonistic mode of action. And there was also a hidden pocket, which we explored, where we could gain potency. And we used QM, and later we have also used the free energy perturbation here to really study the key interactions. It's just illustrating how important this topic is, and the company is now in phase 2B, still progressing towards the market, we hope. So by that, I will just move over to how this platform looks within ASET, just as an illustrative. So important here, the way we work is really through design teams. So in these design teams, we have the chemistry lead, we have the computational expert or experts. So sometimes we bring in multiple depending on skill sets, and also the chemists are there, and also expert from DMPK or assay development, etc., depending on a little bit on the question of the project. So it's really important that all these people with different background and different knowledge in how to do computational chemistry, so to say, would actually have access to all the data, which we have stored in the database based on D360. And then we have also the vision there to everyone should have ability to look at dockings or 3D structures from crystal structures, etc., to really generate ideas. Most of the people we have a PhD, and I see that as a capital loss if we wouldn't use all brains we have and everyone's insights and ideas. So really try to merge that, but it also puts some demand on how this platform needs to look like. So it needs to be easy accessed and also quite easy to handle, but still allowing our experts to play and really work with a more refined way of doing computational chemistry. So for instance, we would like to have the visualization here with the 3D structures, or if that's coming in from cryo-EM structures, we need to integrate that with our group analysis, and also using our machine learning recommender system that should also be integrated, which generates new molecules. And then we should be able to analyze all background data we have there, and then start creating new ideas and enumerate up new compounds on library type ideas there, and then also using 2D sketchers. And then of course, to the bottom here, we also depicted that we need to be able to bring in all our predictive models, which we have built over years from the data we have generated. Also the predictions of 3D structures using different shape dockings, etc., and scoring here to have refined ideas. And then doing some clustering and filtering, and then get that into Make. So for me, it's really important that we spend quite a lot of time here in this design process and use all modeling we can and all predictions, because as soon as we start moving into the Make, that's what really is time consuming and takes long time in the DMTA cycle. So rather more effort here in the designs. And I just want to share a couple of perspectives. Now I can't move forward. Let's see if I can get this to move. Let's see what happened. Oh, I'm sorry, Gabriella. I don't know what happened.
Yeah, I got the same thing, but it unblocked by itself.
Maybe I just stop sharing and share here and see what, if that helps. Okay, let's see. You try. Maybe I just do it this way. Let's see. No, that didn't work. Sorry.
We'll see. Yeah.
Okay.
Yes.
So what I also want to share here is that when you then see the fast progression of new design tools and things we would like to integrate on this platform, it's really rapidly moving. So we would like to bring in our molecular ideation platform, which we have on reInvent using machine learning and AI here, and also the free energy perturbation predictions, which really is a step change with the times where we have a good signal for predicting affinity and combining these. So this is just a couple of these new tools that needs to be on that platform. And then when we have what I also want to point out here is, apart from all the new tools we have for the classical small molecule landscape here, it's also quite a moving field for what we need to have computational chemistry and power for. And that's related to that when we look at the target landscape, there is, as you know, just a small fraction of the genome that is actually coding for proteins. And then if you look at those proteins, it's still quite a small fraction of all the proteome that is bucketed into those kind of more classical target classes where we usually are quite comfortable with doing predictions, etc. And then to the furthest right here, I just looked at one analysis for one of our collaborations with Benevolent AI, generating knowledge graph within the CKD area, and how does that look, right? And there is still a large, great portion of other type of proteins for us to, which is listed as preferred targets to address. So that's a changing landscape where we really try to address these new target classes. And to do that, we have several new modalities there. So We use peptides and oligotherapeutics. And when you think of those, it's not the classical atom depiction, but here to really guide and help the chemist, we need some visualization in 2D sequence, where you can look at amino acids and nucleotide sequences rather than each atom to make some sense of the data. And then we have PROTACs, where you want to really model the tertiary complex and try to understand that, which is also putting a lot of demands on the computational tools you need. And then to the furthest right here, I just want to point out this really interesting area where we're looking into how small molecules could target RNA structures. And as many of you know, that RNA is much more dynamic than proteins, and there's more movements and it's more subtle impact you would have from small molecules to really have that pharmacological effect you would see from RNA binding protein interactions, stabilizing or destabilizing, and then you will have your phenotypic readout. So I'm just wanted to point this out. So despite we put a lot of effort to try to optimize the current platform, it's moving so fast these days around everything new we like to have on it as well. So it's tough to keep up. So happy to have the discussion. Okay, stop there, Gabriella, and leave over to Troy.
Thank you. Thank you very much. I'm going to unmute Troy. Yeah, thanks, Malin.
I am unmuted. Awesome. I'm in charge now. Let me see if I can.
Yeah, let's see if there's any bugs again.
If I can share something here. That should be showing up, I think.
Not yet.
No. You're not seeing anything?
No. It's not our lucky day.
Let's see. That share. Are you seeing space?
Yes.
Beautiful. My son is an astrophotographer, so if you see pictures of space, that's him. Let me start on slide 1. I apologize. So a very, very cool presentation, Malin. I am a chemist, so you're going to get a very different presentation from me. I think Malin's presentation was very top down. Mine is maybe much more bottom up. I've been at Novartis for 20 years. I was a bench chemist. Up until 2020, I was a chemist who very much enjoyed modeling. And being a chemist, you can see here, there's a lot involved in being a chemist. And I think Malin touched on just the amount of time, the huge periods of uninterrupted time required to make molecules. And that huge lump of time required doesn't leave a whole lot of time sometimes for designing, for analyzing the data, for figuring out how to use different tools. So what ends up happening is that a lot of chemists will simply rely on their CAD colleagues to help them model things, or help them come up with ideas, or help them maybe prioritize ideas. And for each chemist, they become very single project centric. There are opportunities, of course, to see what's going on in other projects, but unless you're really able to multitask, it's a challenge to do everything. So let me figure out why my animations aren't working here. I seem to be stuck as well. So if I stop sharing, start sharing, let me see if I can get this to work. Okay, so no, I am stuck. Let me go to here. Try to figure out why I'm stuck here real quick. So let me share that. Share that. Okay, and Bigify. And do we get the second slide?
Yeah, it seems to work.
Awesome. So I think what happened to a lot of us when COVID hit is that we all hit this pajamas period. especially as chemists, trying to figure out how do we work as chemists during the pajama period. But myself, maybe I was in, because I was so intrigued with modeling and had been modeling a lot in my previous projects, CAD at Novartis was very gracious and allowed me to kind of enter their fold. So at this point, I'm very little focused on the how-tos, although I am still a modeler who knows chemistry. This gives me the opportunity to focus more on digital tools, opportunities to analyze the data, design, and be more multi-project centric, maybe too much multi-project centric, in fact. But as Malin kind of pointed on, this pulsing sphere of synthesis is a huge weight on a chemist's time. So the designing, deciding what should we make, maybe more importantly, what should we not make, it's a challenging balance in time. So if we can figure out the best tools to maybe limit the time required for the designing, for the decision making, for determining what should we be making, because the synthesis problem isn't going away anytime soon. So I figured what I would maybe do is pull out these are maybe some quotes in my own head, but these are things I feel like I've heard over the years regarding structure-based design, ligand-based. One of the maybe misconception, having a co-structure will tell us what the final drug should look like. Or if we have a structure, we don't need ligand-based design anymore. Or conversely, if we don't have structures, we can only do ligand-based work, and this is how we're going to operate. Now, if we don't have enough SAR, we can't do QSAR models, we can't do ligand-based design done. What are we going to do? A lot of chemists are like, just tell me the docking scores. Let's prioritize by docking scores. I know that everybody in this room cringes when they hear, I want to prioritize, or I want to overinflate the value of a docking score. And then there's this opinion, which I actually agree with. There are probably too many tools. Many of the tools are way too complex. Modeling is best left to the professionals. I mean, there are companies that have built around this construct. So as a chemist, I wanted to talk a little bit about chemistry. So this is a project that's been presented a couple times before. So I'm not going to show anything that's new or never been seen before. But this is an example that a project that I worked on, the SHP2 allosteric inhibitors, discovery TNO155, which is in phase 1 in combination therapies now for cancer. This is an example in my mind anyway of kind of this yin and yang of, there's structure-based design, there's ligand-based design. If we didn't have both, we would not have made the discoveries we made. So to start off, it's important to know we had a ton of structural data. Every hit that we had, fundamentally, we were able to get structures on. So we had a huge wealth of structural information, multiple ligands all binding in this allosteric pocket in SHP2 in this kind of the center of this structure at the bottom. So we had a lot of chemical diversity. So what should we work on? We had a lot of chemical diversity. Which molecule is the one that we should focus on and then maybe drive toward a lead? We had a large chemistry team, so we were able to work on a lot of different things, but that's really not the most efficient way to go about it. Just work on everything until something fails. You really want to prioritize, right? So here's an example of one of these hit series where we were able to quickly use structure-based design to go from a quite non-potent hit at 12 micromolar. We were able to obtain, like I say, a lot of structures. So we were able to obtain a lot of structures during the process very quickly. So we were able to make decisions based on structure-based decision-making. So one of the things we saw with this hit is that the amino wasn't making any interactions, but that there were some backbone carbonyls very close by. So okay, let's elongate that amino. And in a couple steps, we determined that rigidifying that amino was better. So we ended up in this space where we had extended the amino, able to pick up a backbone carbonyl interaction, but we still had a number of water interactions. And then we were looking at the other end of the molecule, actually the center of the molecule, realized that one of the nitrogens from the pyrimidine was interacting with a water molecule, wasn't doing anything particular with protein, but we recognized that the arginine kind of peripheral hydrogen bond donors were right there, ripe for the grabbing. So we postulated that going from a pyrimidine to a pyrazine would pick up this new interaction, and in fact, it did. So structure-based optimization, in a few steps, we were able to go from a 12 micromolar hit to a 70 nanomolar hit, about 170-fold gain in potency. But this put us in a space of these biaryl moieties, which are they come up as risk flags, but we also identified that yes, indeed, this is a phototoxic molecule, and many of the molecules in this series showed a phototox risk. So what we did was we went back to all of the hits that we had and tried to identify, is there anything that we can pull, any scaffold morphing opportunities that we can take from all of these hits. And what we did is generated a pharmacophore model, a ligand-based pharmacophore model of all of the hits that we had, showing potentially what was driving activity as far as aryls and hydrophobicity and hydrogen bond donors and acceptors. And came to the conclusion that the hydrophobic region here on this left-hand space was being painted as a maybe much larger space than what any one of the ligands were showing, and maybe bigger than what any of the structures were indicating. So we went ahead and incorporated this thioether system into our hit molecule based on this analysis, and found that, you know, breaking up this biaryl system, you no longer have the phototoxicity, and you know, as an added bonus, we gained a little bit of potency there. And then at the amino end, we did a bit of work looking at how the amine had picked up one new interaction, but we were still interacting with a number of waters, and there were a couple carbonyls backbone carbonyls just out of reach. So we hypothesized that extending that amine might increase potency, and in fact it did. And these are all structures; these aren't models. Then we thought, okay, what if we spirocyclize, rigidified the system? Would we be able to maybe increase permeability, or you know, what would be the effect of that? And what we found is we were able to further increase potency with these spirocyclic systems. And this was all designed based on what the structure was telling us to maybe reach out and touch someone, grab some interactions, do something different than what the hits were showing. But that brought us into a space where the structure-based designing kind of came to a halt. The structure kind of told us the shape, it told us, you know, what might work in certain spaces. But that was where we came to a standstill. We had a highly basic system, so we were picking up a huge hERG signal, a number of GPCR signal off targets as well. But we had a very potent molecule. Structure-based design couldn't take us much further. But shifting gears over to more a ligand-based design, looking at what vectors had not been investigated, looking specifically at could we fix the properties of this molecule, decrease the log D, decrease the basicity of that amino, maybe reduce the polarity, the cone of polarity of that amino, and increase permeability. And what we found is that, you know, upping the grease at this end, you still have a hERG signal, you still have some GPR signals. You did get more potency, but you still have a highly basic system here. So simultaneously, we looked at, you know, decreasing basicity in multiple ways, multiple means. We were able to get rid of the hERG signal. So through the ligand-based optimization, and I should say at the other end of the molecule, we knew that we had open vectors, but we didn't have any structural guidance as to what you should put into this solvent space. So that we took that opportunity to maybe improve the properties, decrease the log D of the molecules. So using the ligand-based tactics, we were able to come out of kind of the dead end of our structure-based design into TNO155, where we lost the hERG signal, significant potency, et cetera, et cetera. So I just wanted to maybe talk a moment to each of these misconceptions, that having a co-structure will tell us what the drug looks like. I think we all know in this room that the co-structure is going to maybe guide you how to make a better ligand, but it's not going to necessarily help you getting a drug. Regarding ligand-based design, if you have a structure, I mean, obviously, if you can do property QSAR modeling or designing a better molecule based on property space, that's going to be hugely influential over the drug-like properties of your ultimate molecule. The concept that you don't have enough SAR to do ligand-based design, I mean, all you need is matched pairs. So if you can have a tool that has excellent ability to show you matched pairs with huge differences in potency or properties, that's a huge, huge plus. Docking scores may not be very useful, but property-based prioritization is very productive. So can you look at profile QSAR regarding off targets or regarding improving properties? And like I said, there may be too many tools, maybe too complex, but we have been making progress toward better empowerment of our chemists. And this has been kind of something I've taken up is, can I help my colleagues learn Mo or learn Crescent or learn, learn other tools? Would they be useful for chemists? So one of the concepts that Gabriella is asking, do I have an answer for? What is the most effective way to share ideas? And I don't have an answer for that, because I really have to question, is there a best way? Because every project team that I've ever worked with, different people want different things at different times, and especially depending on the state of the project. But in some cases, a docking score may be very useful. I mean, maybe you have a very small, rigid pocket, and docking scores do a great job telling you or prioritizing your molecules. Sometimes you just want a picture or a holistic view of what does the space look like, and where can I grow into, or where is there a wall that I need to stay out of? Are there existing tools that we can do better? Do we need new tools? And yeah, using structure-based and ligand-based, how can you better interpolate data that to maybe do scaffold morphing, or maybe identify great properties in one molecule to track it over to another, and then the extrapolating as well. I mean, you've got a data set, but where have you not worked yet? To be able to identify that quickly from a structural perspective, from a ligand perspective, look at maybe ligand vectors that hadn't been tried before, or is there a space that goes towards solvent that might be the key place to try to optimize for properties, for example. So the only thing I want to say thank you to all of you guys. Like I say, I'm a chemist. This is my first BAGIM event, so I fully expect that maybe I won't be invited back, but thank you for this time around.
Yeah, thank you, Strug. Thanks a lot. Let me see. So yeah, now the idea is that I bombard you with questions. No, but yes. So we have about 30 minutes left. So let's see how long it will take to go through my questions. So yeah, and for the audience, please type your questions in the chat box. Can I unmute both of you? So but the first questions that where do I start? So you've answered. You've described how a bit about how you work, and I get it that it's very much project dependent of what works best. Maybe I can ask Malin first. Can you comment on how to communicate information coming from 3D, because it's not a score, it's more complex. It's not a property unless it's a docking score, but yeah, we're often looking for a more complex picture than that. So how do you communicate that to the chemists, I guess? Do we need to unmute you again? Sorry.
Yeah, thank you. I think it's a mix. We use Mo and Maestro, Vida, a few different 3D tools. You can store a lot of the dockings in there. I think particularly Vida, I have most experience of myself to be quite fast in like freezing different poses and then sharing those across the design teams, etc. Like you can really go in and click and get the understanding when someone types down in an email or something, look at post there and post there, and you can get the computation chemist's view on what was the thinking and follow it quite easily if you don't happen to sit in the same room. And then we in the design teams, we would play around with these different tools and so on, and also capture ideas. We have used the ChemTracks, which is a tool for just following hypotheses, ideas, and then we type in, this is the hypothesis. You can paste in pictures, etc., and then record your thinking around it and so on, and then later on prioritize if you want to follow up and make it or not, and so on. So I think a lot of the information is stored in the programs, but also captured elsewhere in using the ChemTracks tool, in particular within AZ. So.
Okay, got it. So yeah, so what you're saying is it needs to be processed and or discussed within the team to be able to produce some sort of ranking, right?
Yes, that's how we do it before we would put into the make process. But in between, that could be between a computation chemist or some chemists in a team. It could be the chemistry lead. It could be between chemists and so on, that you have your crystal structure maybe in Vida, and then you would turn it around and so on, zoom in and color code and so on. And then you can freeze different poses, and you can basically store them in the different poses and just click on them. So you would very easy follow what was the thinking around. If then it's more complicated, like you want to look at water networks and anything like that, I think then we always have our computational chemists in there to really understand how really to interpret the data, which water you can move and not, etc. So it's easy to get romanticized around a crystal structure, but it might not tell you the truth. So it's a mix of having it democratized so everyone can play with it, generate ideas, but still having the experts there to rectify if you can't draw those conclusions from what you see.
Right. Okay. Yeah. I'm gonna unmute Troy. Or can you do it yourself? Just to know that how. No, not possible. So do you want to? Yeah, good. Right. Do you want to comment on that same question? How you?
Yeah, I don't feel like we do a great job at figuring out how to share. I mean, from project team to project team, it changes a lot. But you know, some small teams, everybody knows how to use Mo, or everybody's been using ICM Focus. Or, but ultimately, like when the chemists are like, how does this idea look, or what do you think about this? It's always in an email, or it's a ChemDraw, or it's a you know, slides in a PowerPoint, or it's a pasted image in Teams now. So the ideas come from everywhere. And for when, as a chemist, I felt like, hey, this is okay, this works, because I only had to deal with my small team and passing ideas back and forth to a CAD member. But now I'm on the other side of the coin, where I'm working with multiple project teams, and everybody's got different requirements, or different, they want to view in a different fashion, or it's become I really feel for the CAD scientists even more than I did a few years ago, because now I'm beginning to appreciate that if everybody wants something different, it can get frustrating quickly. But you know, in recent days, I mean, I think Rishi's here, but we've been working with Discngine to try to create a platform that is like Google. It should be easy to get into, easy to use. There shouldn't be a lot of buttons. It shouldn't be over complex. That you can get in there, you can look at your data, you can maybe do a ligand-based overlay to identify that if, you know, if this small change makes, you know, what is the effect of this small change? So we're trying to drive into a direction where everybody can ultimately be using the same tools to communicate from chemistry to CAD, CAD back to chemistry. But we're not quite there yet. So it's, every day it feels like it's a different challenge. With, you know, if I share these images in a ChemDraw, if I share these images in a PowerPoint, you're not going to get the full picture. So let's just have a one-on-one for 10 minutes, and I'll share with the team using whichever tool I happen to be using that day, and show them what I think in the space that they're asking about. Yeah, it's a clear challenge. Yeah.
And so do you also track these this explanation or the arguments behind a hypothesis?
We have a chemical annotation and selection tool called CAST that allows us to put molecules in a list, in a table, annotate them with, hey, I think this idea is great, or no, don't do this, do this. And it kind of democratizes the system where people can go in and vote on which ligands, which molecules I like. And then if your ligands fall to the bottom of the list, ideally you never make them, but that's not what happens. So it's a tool, and not every chemistry team uses it, and not every chemistry team that does use it uses it the same. And it's a tool that, again, seems to work better, because if you're looking, it works better for a small molecule project, because when you're looking at tiles of molecules, if you have a small molecule, you can see what they are. But if you're working on PROTACs, you don't know what the heck you're looking at when you're looking at a little tile. So again, project to project, types of molecules to types of molecules, I'm not aware that there is like one best way to do this.
I was also reflecting on what you said there around that is changing, is challenging. I think also the culture aspect here, that we are so used today and so impatient, and especially the younger scientists we recruit are very impatient. They need the information directly, otherwise they have moved away and gone to another idea. So I think that's another challenge when you have to send emails, or like it takes time, or you might not see the person the next or the same very day for discussing that idea. Right. That we probably lose ideas if we don't have that rapid access to information. And I think that's it's not just because of that we always wanted it. I think actually the way we all are starting to demand information sharing is changing as well.
Yeah, absolutely. But by the time we get the answer, they don't care for the answer anymore.
No, if you're not fast enough.
Is that typically people waiting for the CAD or an expert in modeling to be available, or could it be any type of answers?
Based on if I may jump in, based on the number of Teams chat requests I get in a day, I would say that people want answers to their questions immediately. And if Teams have done anything that Teams has done, it's awesome because you can communicate very quickly with everybody, but it's terrible because you can communicate very quickly with everybody. So everybody, if it's in a chat, you feel like it's more urgent than if it's in an email, because in email, a message might sit there for a day before I get back to say, oh, sorry, it took me a minute to get an answer. If it's in a chat, it's almost like it's kicking you, and you need to answer this immediately. So if you get 2 or 3 of those, how do you prioritize? How do you, yeah, wow.
So yeah, Malin, so for you to get an answer right away, is that, so you mentioned that the ability to play around yourself with looking at docking poses and things like that, or what, did you have anything else in mind to tackle that culture, we call it request, or?
No, I think it's just the whole, if you, like Troy described, I think all companies trying to get that ideal design platform, which need to demand easy prediction available. You need to have the 3D structures and different poses quickly. You want to have like all the different tools you need for design. You want them easily. So it shouldn't be too complicated to do it yourself. You want to have, it should be fast. You should have your answer quickly. At the same time, you're putting more and more challenging and demanding computational tools into it. And yeah, what else? There was, no, sorry, I lost one point I had there around it too. But I think it's just in general that we are expecting to have very quickly. Yeah, the last point was actually that all the companies actually built quite refined tools already, and some of them we might like. And then when you get in a new one, it needs to fit with the other infrastructure you have. It's not that you, just because there's one tool that could do it slightly better than your platform right now, is still, if it doesn't dock into your preferred other tools and your database and so on, is still not an option. So I think that modularity to really make an impact is also demanding and challenging for at least AstraZeneca, and I expect it's the same for most companies. You should probably know, Gabriella, as you are talking to the customers.
Exactly. Absolutely. Yeah, people are used to. They are efficient while using things that they know how to use as well, right? So another question. So coming back to the descriptors, and imagine that we have everything centralized in one hub. So we have predicted descriptors of phys-chem properties, things coming from 3D, whatever that, how that is translated into some numbers. How would you imagine the discussion around these different scores? How would you weight different methods in their prioritization? Let's start with Malin.
That was a difficult one.
Sorry.
She didn't prepare us for that one.
Didn't I share that?
Yeah, no, but how would you do? I mean, we have prediction models. It's kind of easy. You can know how well they predict, and then you can also see how they predict across your series of compounds. So you can use, like, which ones would I put more emphasis on, which one would I partly ignore, because I know they are the numbers, but they might not be true. And then, as I pointed out, I think I've seen some really impressive examples when you look at docking and then compare that when we actually have a good free energy perturbation signal for that program and the ligands from that series into the protein. And sometimes they can be really, really useful for us, because if you have, let's say we had a project where we needed to do some scaffold hopping, and you have 30 different scaffold that all would dock in, but we know the complicated chemistry, and then you need to decorate them around to really understand. You can't just do one compound per scaffold, but if you then can use free energy perturbation, for instance, and really rank the scaffolds, and you start feeling that you can rely on that, and you maybe just need to do 5 of those scaffolds and do libraries around them to get into new chemical space. That's revolutionizing in some projects, I would say, from the demand it puts on the chemistry afterwards. So I think it's a discussion with experts from the computational chemists, and also how reliable are the different scorings, and then chemists are same with me in general, skeptical about everything that's predicted. So I think the discussion is there, and it's robust, so as it should be.
Okay, perfect. Yeah. Troy, do you want to comment too?
Yeah,
well?
I think we've made a complete transition in the last couple of years at Novartis, where everybody maybe previously was like, okay, maybe I should calculate some properties for these ideas that I'm coming up with. I think at this point, everybody is running every idea through the in silico properties and models, and getting a reasonable expectation, I think, for maybe not what property space we know we need to be in, but definitely what property space we don't want to be in. So we need, from the ground up, from the chemists, when they're coming up with ideas for targets, they can already pretty quickly say, I'm getting pretty high in my log D, or I'm getting pretty, I've got 5 acids in this molecule. Maybe I need to cut back on something. So it has become that the chemists are coming up with ideas that are better than maybe the ideas that were being come up with 10 years ago. So it's maybe even more of a challenge now to try to filter, as CAD folks, to try to filter through what are the parameters that we need to use to decide to help prioritize. I mean, in many cases for hits, we're maybe trying to prioritize activity, but still maintain a reasonable property space. If you're in a much later stage, or if you have a specific target, anti-target that you're trying to avoid, maybe this is the point where you want to start prioritizing by anti-target modeling, maybe some homology modeling, or some docking in some known anti-target, where you may be able to identify, these molecules are binned as no, these will probably hit that target. But a chemistry team it's like a house that once you've got the chemistry going, it's really hard to move where that chemistry team is and the kind of chemistry that team is working on. That once a foundation of this is what the chemistry team is doing now, it can be really hard to move that house. So we have to do our best to try to identify the best places on the molecules that you can maybe optimize, and that those are going to come from the hard data. Those are going to come from designs, structure-based and ligand-based designing, but it's got to be a data-based decision if it can be. So if there's enough structural data or enough properties data, you have to use that to try to drive your efforts.
I think there's been a slight shift also recently because of virtual screening and AI, which generates not just hundreds of new ideas, it's thousands and thousands, and it's just impractical unless you use all the prediction tools and filters. So we are building kind of refined composite models and you would use your different prediction models with different values, basically given different reliability scores, and then do a composite, and you will use that to refine down, which I think that's used more or less routinely these days because just of the sheer number of compounds generated.
Synthesizability score, right? Just how known is the structure type? Yeah.
So I think it's everything in the society, just because we have much more data and much more computer power, the willingness of using it is also increasing quite rapidly, I would say. So
nice to hear. I'm seeing the time. Thank you for your answers. They're very much on point.
Are there any questions in the chat you want to share?
I think if I start asking them, more will pop in. That's usually how it works. So I'll start with Brian Kelly's question for targeted RNA. So this one's from Malin. Is this something you are or feel can be simulated? How would you approach this? Maybe I guess you can read the question yourself.
Yeah. I really hope there is someone in the audience that knows this better than me, because I also asked the same question. And I think one challenge here is that it's not as well parameterized as proteins. So there's gaps here, which clearly I don't think anyone has the solution to it. But then molecular dynamics would help us to some extent. And then there are a few different programs, Comrades, Shape, and I've been working with Matt Disney on using his databases and so on. It's not really simulation, but there are a few tools, but I think this science is clearly behind from a computational aspect around compared to where we are with proteins. And it's also much more dynamic. And I think the more I understand and think about this area, it's also that it's not, at least my view right now, it's not the same as if you have a protein, you have a pocket, you have a small molecule that binds and you want to compete with a natural ligand in the pocket. It's somewhat static, but if you have an RNA structure, which is quite dynamic, and it's interact with different RNA binding proteins, and then you have a molecule that can bind to that part of structure element to stabilize or destabilize that structure element, which interfere with the RNA binding protein. We're closer to the PROTAC area with tertiary complexes, and how do we, it's quite large complexes to model. It's not as simple, at least in my picture right now, around how to think about it. So I think you need to turn around a little bit in how you think about drug design for RNA versus proteins. I don't have a good answer. If anyone in the audience know how to simulate and model this, I'm more than happy to learn.
Please manifest yourself. Next question then. Thank you, Malin. This one is for Troy. It's from Byron Delabar. One of the difficulties in getting chemists to accept modeling are efforts from previous years that over-promised or overhyped. How does one convince experienced chemists that the tools have improved and have greater utility?
Yeah. Maybe I've been fortunate in my chemist career that I haven't felt like I was overpromised, because I think the best thing that we can all do is be honest with ourselves and say these tools may be able to help us derive better molecules or get into better property space. Or we may be able to generate better ligands through structure-based design. But to be honest and say, we don't know, and we don't know how to calculate what the final outcome should be. I mean, if we could, we'd all be out of a job, and we can't. So I think we have a job for a little while. Yeah, I think the best thing to do is to honestly say, this QSAR model may help us identify what not to make versus what to make, but it's not going to do a great job based on maybe what the dynamic range of the SAR that we have is, or know that there is maybe a 1 kcal per mole error bar here that we may be able to predict one of these molecules is going to be more active than another, but not within a nanomolar to a micromolar range. It's and you also don't want to over-explain sometimes. I think that I've worked with CAD colleagues that have over-explained things, and at the end of the day, a chemist just wants a way to help identify, how should I prioritize the molecules I'm making. And a CAD representative can say, these are the ways that maybe we can prioritize, and then work with the chemistry team and figure out what are they interested, what is the chemical space that they are able to access quickly, and then maybe offer ideas in that space. Because if you if you come back with ideas that are synthetically intractable, or come back with ideas that are out in left field, and like I say, a chemistry team is like a house. If they have to move across the lot, it's not going to happen quickly. So there is an element of sometimes the ideas have to be within the sphere of what the chemistry team can do quickly. And that's a heavy burden on the CAD folks, because we don't always have a good understanding of what can be quickly done. So you have to work with your chemistry team to be able to do that.
And I guess there's it needs to be, you're not supposed to keep the house where it is at all costs, right? Sometimes
you need a little bit of a mobile home.
Yeah.
To be able to move it sometimes.
Exactly. I like the question. Do you have anything to add, Malin, from your experience of overhyped, people not believing in modeling anymore?
No, I think it does change quite a lot, because the tools are getting better and people see that, and also because there are so many ideas coming from different angles, not just about the chemistry team, that also at least our computation chemists use a lot of the AI platforms and like virtual screening tools, etc. So there's a whole lot of new ideas coming into the team as well, and we need to prioritize. And so I think it has really improved, and I think the chemists are using it quite a lot. I even heard a comment from one of my experienced chemists that thought that some of the young ones is coming in and relying too much on predictions and tools. I mean, if it's just a small little pocket you can explore. It shouldn't be too complicated to explore it with your chemistry intuition. You don't need AI always to come up with ideas for a small little pocket. But I think it might be a hype in that direction as well. But it's nice. I think we have a good utility of a lot of tools these days, and so that's my least worry. I think it's just about how to get these tools to crosstalk to each other in a easy, accessible way. And then it will be used.
Yeah. Ideas aren't a problem. It's how to share the ideas effectively.
Yeah. Collaboration. I have another question for Malin. You've mentioned FEP a few times. How reliable do you find the FEP rankings? It's from Christopher Lind.
It varies between projects. I think we have had, a crystal structure is a good starting point. If you don't have that, then I don't know, at least my perception is that it's not much worth, but then it has varied. We have had some proteins that behaved really, really well. But I think you just need to explore it in your series to see how well does it predict. And you can't take one series and then say all series for this protein will work fine. You just need to analyze it. So, but I've seen some really impactful proteins and series where it's worked really, really well. So compared to, at least to where we used to be, and we have ranked, and it has been good fit in getting better compounds.
Thank you. How do I see the question in total? So I have a comment from Vladimir Shupakin. Why are you laughing?
Easily accessible. Easily accessible chemical space is a choice, and you'll often see once a chemistry team has a scaffold with amidability, they'll make amids like crazy, and that would, and what can we do other than amids in the space is the question. So we want to not make 1 million molecules of the same flavor because we can, and that is unfortunately a tendency that we will see with chemistry teams, that sometimes easily accessible chemical space is there, and it sends them down a rabbit hole.
And we're out of questions at this moment.
I think I just want to add there that having strong chemists in your design teams or discussions is really beneficial, because also chemistry is moving quickly now with what is easily accessible. Some of the photochemistry or photoredox chemistry is pretty easy to put on a light in a way to get some rather complex new structures, and then if I exaggerate a bit, and then some under late stage functionalization where you don't need a lot of protection groups and worry about how the functionalization of your compounds are, if you actually can do CH activation directly and so on. And there's a lot of happening in the chemistry space as well, which now opens up new doors for what is ECA chemistry. So I think it's important to have strong chemists that follow the latest literature and dare to explore that as well with you.
Nice remark. I think, since I was aiming for an hour event, I think we will finish the discussion now, unless there are other questions, maybe for a word from the organizers to close the session. Brian, are you still here?
I am still here.
Cool. Hi.
I don't have any questions. I think this has been a great talk, and having done this in past, present, and future lives to organize chemistry software in front of a bunch of chemists, it's a very challenging problem. It's good to hear how people are dealing with it.
Great. Then thank you, dear panelists. Thank you, the organizers. And on the Europe, bravo. Have a very nice day on the other side of Atlantic, and have a nice evening.
And have a very nice evening.
Thanks, Malin. It's great to meet everybody.
Yeah, nice to meet you
everyone.
all. Good luck with your careers and work. Bye.
Bye-bye.
Stay healthy.
Bye.
Yes.